Hello everyone, my name is Renan and I am a PhD student at the Institute of Plant Breeding, Genetics and Genomics at the UGA. Today I will talk to you about how we can increase the protein content in soybean seeds with genomic tools and plant breeding. So, plant breeders have done a great job in increasing soybean yields over the years. However, during this time, the protein levels of the soybean cultivars have been decreasing. To change this situation, we wanted to develop a strategy to ensure that yields continue to increase and the protein levels also increase. A few years ago, our team conducted a GWAS on the USDA soybean germplasm and found a significant region on chromosome 20 that matched previous reports of a high protein QTL in that chromosome. Later, a mapping study identified the presence of a QTL on the chromosome 20 from the parent Dembakong, explaining remarkably 55% of the protein variation. So here, in this research, we aim to identify the candidate gene underlying the protein QTL, develop markers for the selection of the high protein allele, and deploy the high protein allele in the breeding program. Our materials and methods are divided in two parts. In the first part, we used SNP data from the USDA germplasm to identify SNPs within the region of the chromosome 20 QTL and increase the marker resolution in the region. After narrowing the QTL, we sequenced the parents from the original QTL study and some candidate lines identified in the previous GWAS. In the second part, we developed 10 real populations, each one having Benin HP as a common parent. Benin HP or Benin high protein is a near-isogenic line that carries the high protein QTL on chromosome 20. The other parents are elite lines from the southern germplasm. The populations had their protein content determined with near-infrared spectroscopy in 2018 and 2019. Associations between protein and markers were performed using a mixed model approach, in which population effect was fixed and marker effect was random. So let's see some of our results. In figure 3, you can see that the new markers enable a better resolution and the QTL was narrowed to a region of approximately 2.6 megabases. When we aligned the sequence of the candidate lines, we identified several polymorphisms in the region. But the one that got our attention was a large deletion that completely removed the second exon of a gene model. As this was such a strong candidate, we developed a Cas marker based on this deletion. Figure 5 shows that the marker had a good separation of the lines carrying the deletion and the lines without the deletion. In Figure 6, we can see that in both years and across all populations, the marker had a very good prediction of the protein content. We can also observe how effective the allele is in increasing the protein content in all populations. We also wanted to investigate the impact of the allele in the amino acid composition. As you can see in Figure 7, arginine, glutamine, and aspartic acid had the highest increase. There was an overall increase in the content of lysine, cysteine, and methionine. However, the concentration of these amino acids was reduced per unit of protein. The content of tryptophan was not affected by the mutant allele. So, based on these results, we can conclude that the chromosome 20 QTL is effective in increasing the protein, regardless of the genetic background. The fine mapping and sequencing work enabled the development of a functional marker, and the validation study showed that the marker developed can be used for introgression in diverse backgrounds. These populations are currently being advanced to yield trials, and we expect that soon farmers will have access to new cultivars with higher protein content, and we will be able to reverse the trend of protein decrease. Thank you very much.